Hello student, once again welcome back in the digital class of learning. Today we will start the application of animal cell culture in the medical field, or how we are using this animal cell culture in treating the various diseases, which is a type of very chronic diseases. So most of the things which is used by taking the animal cell culture as well as recombinant DNA technology, that is the combination effect, combo effect of the both technique, that is the animal cell culture and RET, recombinant DNA technology. We are applying the protein pharmaceuticals. You know that proteins are basically used in treating the various kind of diseases. So in this heading, that is the application of animal cell culture by using the recombinant DNA technology, what we use, that is the protein pharmaceutical. These protein pharmaceuticals are basically having the medically importance. They have medicinal role. So before coming to this one, you must know that what are the different cell lines are taken out to culture the particular type of proteins or their uses. So first of all, that you have studied about, there are three cell lines which is in your curriculum. Number one is HeLa. HeLa stands for Henebita lax. Henebita lax, that is the HeLa cell lines. Then CHO cell lines. C stands for Chinese, H stands for hamster, and O stands for ovaries. So Chinese, hamster, ovaries. Now again you are having some confusion. What is the hamster here? It is a kind of rodent. It belongs to the class Mammalia. Then cos cell lines. Cos cell stands for cells of simian virus. It is taken out from the monkey and it is a fibroblast type of cells. So these three cell lines we are basically using for the production of this protein pharmaceuticals. And what are the various drugs, what are the medicinal importance by doing the animal cell culture are, firstly EPO. EPO stands for erythropoietin. Erythropoietin is basically used to treatment of the hemophilia, or it is used to the treatment of anoxia and hypoxia. What is the meaning of here? Hypoxia and anoxia. Hypoxia stands for shortage of oxygen. Shortage of oxygen. As we are moving from plain to the hilly region, so there we feel some dizziness or that fainting. So this is because of the shortage of oxygen. And anoxia is the lack of oxygen. So this EPO, that is the erythropoietin. Well, erythropoietin is a kind of glycoprotein hormones, and as I told, it is basically used by treating. It is basically used in the treatment of this oxygen lacking or shortage of oxygen. And more important thing that erythropoietin is nowadays produced with the help of recombinant DNA technology. So we call it recombinant human erythropoietin. This is the name. And this human RHEPO, that is recombinant human erythropoietin hormone, is used basically the hemophilic person, or it is used in the treatment of the anemia. I am not telling about the hemophilia. Hemophilia is something different. That we will discuss in the clotting factor 8 or clotting factor 9. Now next one is that clotting factor 9 and clotting factor 8. You know the hemophilia, it is also known as Beadle's disease. What happens to the hemophilia? A minor cord causes diffuse blood loss or flow of the blood. So basically we are using here what? Clotting factor 8 and clotting factor 8, clotting factor 8 and clotting factor B. Clotting factor 8 is responsible for a type of hemophilia that is known as A type, and clotting factor 9 is responsible for the type of hemophilia that is called the B type. And this factor 9 is also known as Christmas disease. It is also known as Christmas disease. Christmas stands for a patient, that is a 5 years old boy who have suffered first of all, and that disease was invented or it was found in that boy. So it is also known as Christmas disease, and it is known as bleeder's disease. Clotting factor 8, lack of clotting factor 8 is called bleeder disease. Lack of clotting factor 9 is stand for the Christmas disease. Now come to the FSH. Next important is the FSH. It stands for follicle stimulating hormones. You have studied about the biology also. It is a kind of hormones that is basically used in the treatment of infertility. It is basically applied for the treatment of the infertility. Now there are two things: infertility and sterility. Infertility means when the person, that is individual, is not able to produce the ova or the egg, and sterility is something different, both having the different term. Next is the HGH. H stands for the human, G stands for growth, H for hormones. So HGH is used for the disorder related with the growth hormones. It removes the dwarfism. Now TPA stands for tissue plasminogen activator. This tissue plasminogen activator is a kind of proteases enzyme. It is a kind of serine proteases enzyme, and basically used to the remove the clotting of blood in the vessels or in the heart. So this TPA is used in the cardiac attack or cardiac arrest or the heart stroke, because sometimes the blood clots in the blood vessels or the chamber of the heart. So it is the first drug which is given to the patient so that the blood can dissolve. It will be discussed later on just now. Now IL-2, it is stand for interleukin 2. Interleukin 2 is responsible for the treatment of the cancer. We are using this IL-2 in the treatment of the cancer. And apart from that, another thing is that MAPS. MAPS stands for monoclonal antibodies. Monoclonal antibodies is antibodies. It is artificial antibodies that is used by the hybridoma technology. We will discuss on later. And it is used in the treatment of the cancer cell. So what are the different cell lines we are taking here? This TPO, clotting factor 8, clotting factor 9, FSH, HGH, TPA, IL-2. These are developed by using the CHO cell line, that is the Chinese hamster ovaries cell line. And this MAPS, what kind of cell lines we are using here? Hybridoma cell lines we are using. So now come to the tissue plasminogen activator in detail. Tissue plasminogen is a enzyme and it belongs to the serine proteases. It is the category's name is the serine proteases. You have studied about the clotting of blood. You know the clotting of blood is a multi-step process where fibrinogen is converted into fibrin and the fibrin is responsible for the clotting of the blood. But here what happened? This plasmin is responsible for dissolving of the clot blood, and more importantly, suppose this is a plasminogen which is an inactivated form of the enzyme. It is converted into the plasmin and thereafter plasmin to fibrin, and these fibrin when treated with the TPA, that is tissue plasminogen activator, then hence what happened? The blood cells dissolve. So whenever it is injected into the body, wherever the clotting of blood takes place, it will be dissolved. Now how it is used nowadays? By applying the recombinant DNA technology. First of all, we will take the plasmid. A plasmid of interest has been taken out there. The same process we are applying here. Now cutting the plasmid from the desired place. Here it is cut out here. Now we are pasting here the TPA gene. TPA gene is pasted here or it is inserted here. You can see here. After the insertion of TPA gene on the relevant plasmid, it is introduced into the mammalian cells or it is inserted in the mammalian cells. Now what happened? These number of mammalian cells are there. These mammalian cells put in the matrices and they are now able to form the TPA. And these TPA, when applying or when they are given to the patient, it works without harming the other cells. Similarly, one more important thing I forget about the EPO also. Actually, EPO is the one of the very important and non-risky medical facilities. Normally, when the person is lacking from the hemoglobin or RBCs, there is a blood transfusion taking place, and sometimes this blood transfusion also giving some diseases or they spread the diseases in a healthy person. But when this EPO, erythropoietin hormones, are directly inserted into the human cells, so there is no harming of such sorts of the diseases. So once again, these animal cell cultures are used in the treatment of the various kind of diseases, which is protein pharma based, and we are applying or we are taking a number of cell lines. The cell lines are HeLa cell line, CHO cell lines, COS cell lines, and EPO, clotting factor 9, clotting factor 8, FSH, HGF, TPA, IL2, etc. Here we are using the CHO cell lines, but MAPS, monoclonal antibodies, it is anti-cancerous drugs. Here we are applying the hybridoma cell lines. And I just told about that. Recombinant human EPO is basically used to treatment of the anoxia and hypoxia. It is related with the anemia. Anemia is the lack of hemoglobin or lack of blood in the cells. And more importantly, nowadays we are applying a TPA, that is the tissue plasminogen activator. It is a group of the serine proteases and it is used to dissolve the blood clots and giving to the patient who are suffering from the cardiac arrest. Thanks for watching.